Okay, afternoon, everyone. There's still very few of us in the room, but I think we should start. Just for those of you that don't know me, I'm Sheriff Geertz and I'm the South African Association of Botanists Council. Glennis, our chairperson, couldn't make it today. She's in the field. So I'm just standing in. Before we start, just in terms of Sakna's points again, please feel free to put your name and surname in the chat, indicating as such, and we'll pick that up. Yeah, so it's my great pleasure then to welcome our guest speaker for today, Steve Keller, and we appreciate, Steve, that you want to tell us about your research and already genomics can predict population vulnerability to maladaptation under environmental change. So just some background. Steve's lab researches the interaction between genomic variation in natural populations and environmental change. And he has a focus on how human impacts on the environment, so particular climate change and biological invasion, and how that alters the genetic and ecological context within which species evolve. His primary studies on the genetics of forest trees and invasive plants. So some background on Steve. He got his bachelor degrees, Bachelor of Science degree in Pennsylvania, the Juniata College, and then he moved on to the National Park Service in Colorado. And from there, he ventured north to do a master's at the University of Alaska Fairbanks, where he studied breeding system evolution in Arctic alpine plants. Then Steve got his PhD at the University of Virginia with Dr. Keller on the population genetics of invasive species. And then he did a postdoc at the University of Minnesota in Peter Tiffin's lab, working on population genomics in populace. Currently, he's at the Department of Plant Biology at the University of Vermont. Steve, yeah, thanks a lot for sharing your research, and we're really looking forward to your presentation. And with that, I would like to hand over to you. Thank you very much.
Great, thanks so much. Can you hear me okay?
Yes, we can hear you, and we can see your slide. Thanks.
And the laser pointer too.
Oh yes, yeah, there we go. Yes, I can see that. Thank you.
Great, wonderful. Well, thank you so much for that introduction and for the invitation to come and speak with you and share my work. Very grateful for the opportunity. So my lab, as was just mentioned, focuses on these two components of anthropogenic change and how they're impacting the world's biodiversity. So we know that we've been moving around the world's biota at an unprecedented rate and causing many different species to be introduced to areas where they really have no native history. And these often result in biological evasions. And so we can think of this as sort of spatial anthropogenic change in the sense that, we're moving organisms around into new biogeographic regions. And then, of course, we know that we're also undergoing a temporal component of anthropogenic change in the form of a changing climate. And as you can see in the graph here, the last decades have been the warmest on record since we've been recording the climate. And we know that as discussions are happening in Glasgow right now among the world's nations on how we might mitigate some of the warming that's already underway, what's clear is that warming is happening and it's ongoing, and that the effects are going to be felt within the short to medium term time frame, right? So even if we get our act together and start to curb emissions, we're still looking at increases of 1.5 to 2 degrees C above baseline levels. And many of those changes are going to impact organisms within their lifetimes, long-lived organisms, including ourselves, but also many plants that have generation times that will span this period of change. And so it's really imperative that we begin to understand and be able to forecast what the impacts of this short-term climate warming is likely to be on the world's plants, and how we might mitigate those changes. If you're familiar with type of climate forecasting called species distribution models, this has sort of been the one of the main tools that biologists have used to try and predict, particularly how species ranges might be affected by warming climate. And so in species distribution models, or SDMs for short, basically what happens is you take a set of occurrences, perhaps from herbaria records or other online databases like the Global Biodiversity Information Facility, GBIF. And you intersect those occurrence points, those latitude and longitudes, with climate data, gridded climate data, to build an envelope that describes the species ecological niche in relation to climate. And so I'm showing you one of these SDMs for species that my lab works on, red spruce here, which is a cool montane coniferous tree. And what you can see here is that under current climate, you've got a model of where you expect to see the species just based on its climate relationships. And then once you have that model, you can make a forecast or a projection under future climate, for example, in 2090, and then you can make a prediction about how the species range should shift, and then sort of look at what parts of the range are likely to show extirpation or loss of suitable habitat, and then where you might gain some habitat into the future. And so these models are useful. They're inherently correlative, but they've seen a lot of widespread adoption, and they can yield some interesting insights, but they also have limitations. And one of their key limitations, in my opinion, is that they ignore variation within the species, so-called intraspecific variation. So they basically treat the species as homogeneous in terms of its response to a change in the environment. But we know that populations within a species show intraspecific variation for key functional traits, right? Like the timing of their growth, phenology, or their ability to tolerate abiotic stress, like drought or high temperatures. And so we know that local adaptation within a species range is commonplace, and there's abundant literature on this, particularly in plants from evolutionary ecology. And so what that means is that when we're thinking about climate forecasting and how that might roll into, predicting risk of maladaptation, what we really need to do is think about how along an environment, the phenotype or the trait of a species of interest may actually be genetically locally adapted to different portions of that climate gradient, which means that as we forecast the climates of the future, what we really need to think about is where within each of these different populations they may experience a shift in the new optimum. And the old phenotype may no longer be locally adapted under a future environment. And so we need to be thinking about this more at the population level within a species. And so the one approach to this forecasting is to use genomics. And in genomics, typically what we would do is we'd go out and we'd get a sample of our species of interest from many different locations, like those populations arrayed along a gradient. And we'd extract DNA and use high throughput sequencing, so-called next generation sequencing, to identify many individual variants or single nucleotide polymorphisms shown here throughout the genome. And one of the benefits of being able to do this is the costs are actually coming down quite a bit. And the ability to sequence is no longer the domain of just a few model organisms, but you can pretty much sequence any species now. And once you have those data, you can relate the variation in the genetics to here the genotype at each of these variants to some aspect of the environment that they came from, and determine the association between genes and the environment, perhaps that underlies that local adaptation that we were talking about. And you can also relate the phenotype itself to those gradients, for example, using common garden studies where you grow many representatives of these populations under common environmental conditions and measure how their phenotypes reflect the environments that they've evolved under in their source. And then you can also, in the third axis here, look at how the genotypes at the molecular level might be able to predict some of these locally adapted phenotypes. And so we can think of this as sort of a triangle relationship of genotype, phenotype, and environment. And if we can understand these three axes that underlie local adaptation, then we may be able to make some useful predictions. And so the question that I've been thinking about a lot lately and working with collaborators on is, can we use this framework of ecological genomics to predict maladaptation under changing environments? And the idea is that basically populations should be at a locally adapted peak of high fitness relative to their environment. So that if the environment shifts, and you have sort of a moving target, then how much maladaptation would you expect or would you predict given the amount of shift in the environment? And you can see that nicely in the animation over here, where if the fitness peak is shifting because the climate is changing, then how abruptly does that affect the populations, and how quickly are they able to reconnect either through various evolutionary processes such as migration and selection to reconnect themselves with that fitness peak. So how do we go about this? Well, first, we need to discover the regions that show local adaptation within the genome. And so we would go out and acquire some data. We would sequence, like I mentioned previously, genomics from a diverse collection of samples. And ideally, we would pair that with phenotypic data on how these plants are growing and what their stress responses might be. And then also get high resolution climate data for these same samples. So you have these sort of three axes of genotype, phenotype, and environment. And then there's a well-established body of statistical techniques in population genetics that can relate the genomic data to predictors of environment or how genomics may predict phenotype. And so these are known as gene environment association scans, genome-wide association studies, or FST, genetic divergence metrics of local adaptation. So all of these analyses, it's not important that you are deeply familiar with any one of them, but they're different approaches towards finding these peaks of interesting genetic variants that may suggest there's a set of genes in that portion of the genome that underlies local adaptation and the response to the environment. So once you know or have some candidates for what the genetic basis of that local adaptation is within the genome, then you can begin to generalize those relationships and make some predictions in space and map the turnover of the adaptive component of the genome along environment here. So if we have some function that relates how genetics is impacted by environment, then we can map that in space and see how different portions within a species range may actually vary in their genetic composition. And then importantly, once we have a general model like this, we can make projections of how the optimum might shift under a change in environment. And that's really the basis of what we're trying to get to here. So if we understand the current pattern of adaptation, then can we make a prediction about how much population might be shifted off its fitness peak if the environment changes? So my collaborator, Matt Fitzpatrick, and I came at this problem, and we were thinking about it for a while, and we thought that the current approaches to being able to do this had some limitations. And one of the big limitations is that they were treating each particular gene locus individually and trying to analyze, one gene at a time, basically. But the genome it's a complex system, and most traits that we care about are highly polygenic, right? They're made of, or they're composed of many genes, each contributing a small impact to the overall extent of adaptation. And so we decided one way to approach this problem would be to borrow methods from community ecology. And in community ecology, biologists are often interested in modeling all of the species within a community that makes up the biodiversity of that particular community. And so many different species in different abundances. And so we said, well, we could do this same approach, but instead of many species, we can look at many different genetic variants across the genome, treating the genome essentially as a community, and all of the different gene loci within the genome as the different members of that community. And so in this approach, we took an existing method called gradient forest, which is used in community ecology to model the composition, the biological composition of a community. And we extended this to handle genetic data. And so gradient forest is a non-parametric machine learning algorithm that's basically built on the backbone of random forests. And it's described originally in this paper by Ellis et al. 2012. And the basic approach of gradient forest is to take genomic data from many populations that you sample on the landscape for many different gene regions. So you've got allele 1, allele 2, and so on for many hundreds of thousands or millions of loci. And the data matrix here is simply the frequencies of those alleles in each population. And we can model those frequencies as a function of many different environmental characteristics that we can measure at each of those population sites from which the samples came from. And so these environments might be different aspects of climate, such as growing season temperature, or maybe the maximum temperature experienced during the height of the warm months, or they could be different gradients of precipitation or soils, or even biotic variables such as pathogen infection rates or herbivory. And in gradient forest functions, the algorithm tries to determine how allele frequencies split along some environmental gradient. And so it starts with the entire set of allele frequencies, and it first tries to split the entire pool into two different groups based on some threshold that defines a difference in allele frequency. So this could be, a temperature, a portion along the temperature gradient that distinguishes a population that has a high allele frequency versus a low one. And it does that iteratively, trying to split the allele frequency data along the environmental gradients in a way that best explains the differences in allele frequencies seen between populations along the landscape. And the sum of these splits can be visualized here as a cumulative importance curve along the environmental gradient of interest. And so the way you interpret these curves is along this portion of the gradient where the curve is relatively flat, most populations have very similar allele frequencies. And then as you start to move along the gradient, you get more and more of these splits happening that distinguish populations on either side of a portion of this gradient. And as those splits accumulate, then the overall importance of the curve increases. And it increases monotonically, so it only goes up. And you can get some interesting information from these turnover curves, but the shape of the curve suggests the steepness or the rate of change at which the alleles are changing across the landscape. And so we see the most allele frequency change along this portion of the gradient between 12 and 16 degrees C, whereas there's relatively little change in allele frequency at the very high end, say after 16 degrees C, and at the very low end, up to maybe about 11 degrees. And then the overall height of the curve suggests the overall importance of this particular environmental gradient in explaining genomic patterns in allele frequencies. So we originally described this method using genomic data from balsam poplar, which is a boreal tree in North America, widespread throughout Canada and into Alaska. And so one of the genes that we knew affects phenotypes in terms of leaf out and flowering and dormancy transitions in poplar is this circadian clock gene, gigantea. And the map here is showing allele frequencies at a given SNP within gigantea across a large set of populations. And so you can see that the ancestral allele here in red is relatively high frequency in the eastern part of the range. And as you grade to the northwest, you have this gradient in allele frequencies. And so the turnover curves here show individual SNPs within this gigantea gene highlighted in red here. And so you can see the very strong turnover happening along gradients of summer temperature, suggesting that this gene is responding, has been selected to locally evolve under different conditions of this temperature gradient. And so the gradient force captures the signal in these data, and we can use it then to make projections. And so those projections, what we really want to know is how much the gene environment relationship might get disrupted from its current local optima if the environment changes, and we can formulate that into an expectation of maladaptation. And so we call this expectation genetic offset. And genetic offset is simply the difference in the y-axis here, the adaptive genetic component of turnover, given a population's current position along a climatic gradient. And then if the climate were to shift, say increase in temperature by 2 degrees C, where would it then put the population along that turnover function? And so you can see for population A here, would require a very large shift along a steep part of the gradient. And we can quantify the extent of that shift here as a genetic offset, which is just the difference in adaptive turnover from its current position to where it would be expected to be given a change in environment. So for population A here, we see a large predicted genetic offset. But for another population whose current position is on a different part of the gradient, a similar increase in 2 degrees C might only have a small shift along the adaptive genetic turnover gradient. And so we see a small offset here. So what this lets us do is make population-specific predictions about how a given amount of climate change might affect a population based on its genetics and the relationship between genetics and the environment. And you can think of this genetic offset concept as an in situ disruption of the gene climate association. So for any given population within the landscape, it it inhabits a space, a longitude and a latitude at its given time point, and that space is under current climate conditions. But if the environment changes and the species doesn't move, doesn't migrate or disperse to try and track that change, but it instead stays in place, how much maladaptation would we expect to see under the new time point? That's the concept of genetic offset. So how maladapted would this population be given its exposure to climate change in its current location? And so we can estimate genetic offsets in a spatially explicit way for every location on the landscape within a species range that we have high density climate data for, using this turnover function that we estimate from the genetic data. And so for balsam poplar, for example, you can see this is a genetic offset prediction map based on the 2050 projection of how climate is expected to change in North America. And you can see examples of those two types of populations, A and B, where A is existing in a part of the range, these warmer colors, where the expected turnover is likely to be high, and hence its genetic offset is expected to be high. Whereas population B here is an example, it's going to experience a certain amount of climate change too, but its location along the genetic turnover function makes it less predisposed, less vulnerable to maladaptation. And conceptually, this idea of genetic offset is closely related to climate transfer distances that have been used by foresters for many decades. And in forestry, it's common to sample many seed sources, so-called provenances, shown here by these circle points, from across a large portion of a species range, and then plant those provenances into trials or plantations at a given number of sites, shown here in the triangles. And so you're essentially transferring through space, not time, a population from its local climate into a test site. And that concept of a climate transfer distance, the foresters have used to measure how growth of the different provenances varies according to how far away they were from their source climate. And you can see that there's this sort of expected stabilizing selection. If the populations are locally adapted, then the performance is going to be at its peak or near its peak when the test site climate is very close to the climate of origin. And that's essentially what genetic offset is saying too, that instead of growing the resources needed to grow many trees for a long number of years, often many decades in these provenance trials, genetic offset says we can approach the problem similarly using genomic data and potentially be able to address questions that couldn't be addressed on a shorter time scale without the need for these long-term common garden studies. So since we introduced the genetic offset concept in 2015 in the original paper, a growing number of studies have begun to implement and estimate genetic offsets for a wide variety of species, including lots of forest trees, which is sort of how we originally began thinking about genetic offset, but also for a wide variety of other plants and other organisms too, including birds, bats, goats, and a variety of crops too. And this is a table taken from recent annual review in ecology, evolution, and systematics that Thibaut Caplanc, a postdoc in my lab, led the writing of. So if you're looking for a good introduction to the genetic offset idea and some background, I recommend this paper. So we wanted to know, as the concept was getting adopted and more studies were beginning to estimate genetic offsets and suggest that they be used for making conservation decisions, we wanted to get some simulation testing of the idea to see how well it actually works in predicting maladaptation. Because right now, this was all prediction and we didn't really have any validation. And so I want to share a few slides from a simulation study that's on bioRxiv right now, led by Alki Laursen, who is a former postdoc in Katie Lauderhaus's lab. And this is also in collaboration with Ben Haller at Cornell and Matt Fitzpatrick at University of Maryland. And so the idea here was to use truth-known simulations of population genetic data in a genetic offset framework to see how well it predicts the actual loss of fitness when you transfer a population into a novel environment, to see whether the algorithm was actually giving us an accurate view and potentially where some of its shortcomings might be. And so in the simulation here, we have, it's a two-dimensional set of populations. So each circle is a population, and it's a 10 by 10 grid. And populations exchange genes with each other through migration between adjacent genes. And we explored both single locus and multi-locus models of adaptation and looked at both simple and complex environmental gradients that these populations were adapting to, including knowing the causal environment that was creating selection on the genome, but also several fake environments that were partially correlated with the causal one, in addition to several other environmental predictors that weren't correlated. So the idea here is really to see how well gradient forest is, how well it does at picking out the actual causal environment driving the adaptation and be able to distinguish it from the sort of noise of some of these other correlated variables. Because if you've ever worked with climate data, you know that many of the variables show a high degree of correlation with each other, but that doesn't always mean that they're all causally related to the phenotype of interest. And so sort of cutting quickly to the results, the single locus model, where there's a single locus that's under selection and responding to local adaptation along the environment, we see that when we look at how a population's fitness does in response to getting transferred from its home environment into a novel environment of a common garden within this landscape, we see that the genetic offset predicted by gradient forest does a pretty good job at predicting the fitness of individuals upon that transfer. And the steepness of this relationship, they're all significant, but we found that it varies somewhat depending on the location of where the common garden experiment takes place. So the locations 1 and 10 are transferring populations to the far left or the far right extreme side of the gradient, whereas population, the common garden location of 5 is transplanting populations into the middle of the range, basically. And so we see a steeper relationship, as you might expect, if you were to do the more extreme transfer to the sort of edge of the landscape, which represents some of the most severe genetic offset transfer distances. We also see that gradient forest does a pretty good job at picking out the true causal environment underlying that selection, and that the fake environment that shows partial correlation in our simulations with the causal environment, as well as some of these other environments, show relatively low importance. So the algorithm's doing a good job at picking out the environment of interest. But we did discover that gradient forest is sensitive to declines or gradients in population size, even if there's no selection. And so these figures are showing an example of where a purely neutral model with no selection in it, how gradient forest cumulative importance changes along the environmental gradient for either a gradient in decreasing population size. So when the populations on the far left of the landscape are large and then become increasingly smaller to the right. And then we also did the opposite or flipped scenario where the populations on the far left are small and increase to the right. And so this is meant to simulate what might be changes in population size within a species range as you get towards from the core to the margin of the species distribution, where you expect genetic drift might increase. And so what we see is the algorithm is sensitive to these changes in population size, which reflect the magnitude of genetic drift going on. And in fact, deemed size shows a relationship with the genetic offset metric. So this sort of underscores the need to very carefully verify that the data you're working with are truly under local adaptation and not just subject to genetic drift, and to be particularly careful in situations where the environment itself may also co-vary with the size of the population. And then turning to the multilocus model, we see genetic offset does a pretty good job across a range of different cases where the environments may vary both from horizontally and vertically in their gradients within the landscape, and even where the environment has an intermediate optimum within the landscape. So we did different case studies varying the spatial arrangement of the environments. And we can see in these warmer colors here that genetic offsets consistently show a strong negative correlation between the magnitude of genetic offset and the degree of fitness, as predicted if they are doing a good job at predicting risk of maladaptation. Interestingly, if you just use the environmental data on their own, then if you know going into the scenario what the causal environment is, then the environment alone does a pretty good job at predicting loss of fitness. But if you don't know the causal environment and you instead include all of the environments that you're measuring, then we can see that it does a much poorer job of being able to predict fitness under environmental change. And then this is just the multilocus measure of adaptation. And so high degree of genetic offset under a multilocus model of polygenic adaptation shows a strong negative relationship with common garden fitness. So all of this suggests that there's a pretty good basis for being optimistic about using offsets to predict maladaptation risk. But we wanted to move out of simulation world and try and validate this concept in nature using a couple of different case studies. And so the first case study I want to show you is for balsam poplar, that species that we originally developed genetic offset ideas around. And here we're using common gardens, similar to forestry plantation trials, to move populations taken from the north and the south of its range. We're going to move these into two different common gardens, one in Vermont near the University of Vermont where I work, and one here in Indian Head, Saskatchewan. And so the genetic data here we obtained using genotype by sequencing approaches for 42 populations, shown here in red points. And we obtained SNP data for a little over 100,000 SNPs for each of these individuals. And then used a range of different local adaptation genome scan approaches to detect which of these SNPs showed strong signals of local adaptation and could be used to estimate genetic offsets. And so here's an example of the cumulative importance curves along a latitudinal gradient based on these results. And I'm showing you different curves for different sets of climate outliers determined using different methods for detecting local adaptation within the genome. And you can see they basically all show a very similar pattern of turnover, although they differ a bit in their sensitivity of the total amount of importance they can explain. And we also wanted to see what the part of the genome that wasn't necessarily showing signals of selection might also be turning over along these gradients. And so we selected some SNPs randomly from the background and estimated turnover functions for this random genomic background. And you can see that indeed the genome as a whole does show some relationship, although it's much less strong compared to the adaptive SNPs. We can do this for all of our different climate gradients simultaneously in a multivariate model to identify which of these climate gradients seems to be most important in driving the pattern of local adaptation. And you can see that they become increasingly less important as you move from left to right through the figure here. So latitude, winter temperature, summer precipitation, and winter precipitation are the most important in structuring the genomic data. And so we can use those turnover functions to calculate genetic offsets, but instead of calculating them through time, we calculate them through space upon transferring from a population source climate to the climate located in the Vermont Common Garden here. And so the size of the circle here is proportional to the magnitude of genetic offsets. So populations that come from nearby the Vermont Common Garden site tend to have relatively low offsets. And you can see populations with larger offsets over here. And we can, just to underscore that this is really a genetically informed metric, we can contrast the genetic offset magnitudes for each population with the raw climate distance with no genetic data underlying it. And we can see that you get a different set of predictions. For example, these populations here have relatively low climate distance to the Vermont Common Garden site, but relatively high genetic offset. So how well do these different predictions correlate to a metric of performance within the Common Garden, which here we're going to use as the height that the trees grew over the course of the experiment? So if we look at just the raw climate distance with no genetic data in it, we see a significant relationship. It explains about 34% of the variance in height, but there's quite a bit of noise in the data still. So climate can predict growth, but does so imprecisely. Whereas if we calculate genetic offset functions, then we see that those are much better predictors of the amount of growth that a tree is able to put on in the Common Garden, explaining over 60% of the variation in height. Interestingly, so this was for the candidate SNPs that we identified using the genome scan methods. If we do the same thing for the random SNPs from the background of the genome, we see a similarly strong relationship. And we think this probably reflects the fact that similar to isolation by distance effects on population divergence and allele frequencies, isolation by environment can also similarly structure gene flow between populations across the genome, not just at the selected SNPs. I want to show you a second case study from Vermont that focuses on red spruce. So red spruce is a mountaintop coniferous species adapted to cool, moist climates, shown here, looking out from the summit of Mount Mansfield in Vermont. And in red spruce, we collected populations from throughout its range here and used genetics to identify 1.4 million SNPs. And we see that there's a fair amount of population structure shown here in this genetic PCA in red spruce. And the population structure varies within the range into this core group up north, a marginal group in the transition zone in Pennsylvania here, and then these edge populations as you move into the fragmented portion of the range further south. And these populations inhabit very different climates, where the core of the range is cool with a large amount of precipitation as snow, and the edge of the range has a long frost-free period and high potential evapotranspiration and very warm growing degree days. So we've got sort of a climate gradient going here from north to south. And similar to the poplar experiment, we transferred and grew seed from each of these source populations in 3 common gardens. And this is work that was led by the PhD student Anu Prakash in my lab. And using the genomic data, those 1.4 million SNPs, Thibault was able to identify genes that were significantly correlated along the source climates that we sampled each of these populations from. And I'm showing you the allele frequency change across the gradient for just 6 of these genes that he identified as being under strong local adaptation. So putting these 2 data sets together, the phenotype data from the common gardens, and then genetic offset predictions from the genomic data, we can see that for red spruce, height growth in the gardens is, again, very well predicted by the genomic offset. So high genomic offsets over here tend to be associated with low growth in the gardens, and populations that show low genomic offset tend to show better performance. And these are data that another postdoc on the project, Suzanne Lockmuth, has been instrumental in developing. So what can we use now that we've shown that the genomic offsets can be validated using empirical data? What might we use them for? Well, one would be to design conservation approaches such as assisted migration or assisted gene flow, where we want to know what are the sources that will be best able to deal with the environments of the future, and then target those locations to collect seed for restoration. And so to get at this idea, we had to think about genetic offset in a slightly different way. And Andy Goerty, another postdoc on the project, came up with this idea of a reverse offset, which predicts pre-adaptation of existing populations to a future climate. And so here, the concept of reverse offset can be thought of as within a given spot within the range, if we look at the climate predicted for that location in the future, maybe it's a bit warmer than it is now, can we find populations currently that would be a good match to that future climate at this future location? And so we can look for a match to this future climate based on where in the range currently we might find population that is well adapted. And so this is the idea of the reverse offset. And we can do that for many different locations and try and find where within the current range maladaptation will be minimized and might be useful to select for restoration, planting into a given location in the future. And so essentially we're asking how pre-adaptation pre-adapted our existing populations to future climate within a given target location. And so Suzanne took this idea and applied it to the red spruce case study that I showed you a minute ago. And you can see for a couple of different time periods and 2 different forecasts, the colors here show the extent of genetic offset predicted under the reverse offset metric. And the triangle here represents a potential planting site that we might want to establish reforestation in. And then the colors reflect the appropriateness, or where to minimize reverse offset, where you might look. So you might look for populations along the coast here, which would be the best match for the current, for the future climate for this region here. And in particular, those fragmented populations down south, there's a large amount of restoration concern and effort among these populations. And so we've already been working with collaborators involved in the private sector conservation communities to identify optimal source populations for restoration sites under future climates. So now that we've shown some case studies, and it's beginning to get increasingly adopted, this genetic offset method, there have been a couple of important papers that have come out recently that highlight both the opportunities that genetic offset has, but also the challenge that we have in assessing their appropriateness, and in particular, validating the concept with experimental data. And so I want to talk at the end of the talk here, just a few points about what assumptions does the offset metric make, and what caution should that inspire in us before we're ready to implement these ideas into conservation. So one assumption is that populations on the landscape right now are currently at their locally adapted fitness peak. And that may not always hold. For example, many species show a lag following climate change at the end of the last ice age, particularly in temperate latitudes, that may cause an adaptation lag following range expansion. And there's a nice study that was recently published in Proceedings of the National Academy of Sciences on valley oak that showed the current pattern of local adaptation that you would predict on a climate transfer to a new site is quite a bit different than what you actually see in populations, which is indicative of an adaptation lag. In other words, plants are still trying in valley oak to catch up to their optimal climate. And the relative growth under common gardens isn't maximized at the climate that is most similar to where they are now. It's actually maximized in an area that's different from their current climate. So this is an important consideration, that populations may not always be at their current fitness peak. Another assumption is that allele frequencies are only increasing monotonically along the environmental gradient. But might there be cases where allele frequencies don't increase monotonically? For example, maybe show a quadratic relationship along an environmental gradient, such that high allele frequencies might be predicted in the middle of the gradient, but at either extreme of the gradient, perhaps low allele frequencies would be predicted for that gene. And we still don't quite know how important non-monotonicity might be in real genomes, but it is something that all of the current methods basically assume and needs to be further investigated. And then we saw in the simulation data that neutral demography in the absence of selection might have very large impacts on genetic change in allele frequencies through space, and hence will also impact genetic offset metrics. And so the assumption that a change in allele frequency along an environmental gradient is due to selection and not genetic drift or other features of demography is a very important underlying assumption in the method. And it really underscores that only selected loci should be used to train the model, and that we have to be careful that genome scan procedures that identify those candidate loci correct for demography. Then lastly, like any modeling, extrapolation beyond the range that the model was trained on is inherently risky and probably not a good idea. And this is not necessarily an assumption, but it's worth bringing up that, you know, if we have a metric along a climate gradient of relative fitness that we're using to estimate genetic offset, we can't really extend beyond the climates that we have available to measure right now, because we don't know what the relationship might look like beyond that, the known range of environmental data. And this is particularly an important problem for climate change, where some aspects within a species range might extend beyond the current climate gradient that we are able to measure. And we don't really know how the population may respond to that. So in closing, future directions and prospects, where do we think genetic offsets might be going for the future and how they might be used? Well, we talked mainly about this gradient forest approach, but there are multiple different approaches to estimating genetic offsets. And a next step is going to be to rigorously compare and evaluate these different metrics, and hopefully develop and refine new ones as the community of researchers continues to push this field forward. And I think simulation has shown a lot of promise in being able to understand how the algorithms work and what aspects of sampling and experimental design and even genetic architecture have on genetic offset predictions and how well the models perform. And then I think it's very important that we explore new validation approaches and the ability to predict fitness. And the common gardens are a good example, but I think we could also explore novel approaches like experimental evolution for those systems that are amenable to it, resurrection studies where you can compare seeds, for example, that have been stockpiled from an earlier time period and grow them concordantly with modern material, and even dendrochronology studies, like in trees, where we can relate tree rings to genetic offset predictions. And then I think there are a lot of potential new applications for this idea. One that hasn't been touched, to my knowledge, is its application in biological invasions, where the transfer to a new environment is primarily through space and not time. And I think there's also a great potential to use these ideas in agriculture to find optimal areas to grow crops under new climates. So I've got many collaborators that have contributed to this work, and I've tried to highlight their contributions along the way. But I appreciate all of the folks here, and also funding from the National Science Foundation, USDA Forest Service, and the University of Vermont. And I'd be happy to take any questions. Thank you.
Thank you so much for this wonderful talk, combining genetics and climate change, and explaining genetic offsets, but also reverse offsets in such a clear and logical way that I'm not a geneticist can follow and understand. So thank you so much. So let's open the floor for questions. You can just raise your hand and then unmute and ask your questions. If you don't want to ask in person, then you can put them in the chat. But I prefer if people just speak up. I don't want to abuse my position as chair, but whilst we wait for people to to ask questions, I have a few that I really want to ask. Yeah, so there's a few that's on biotic things, but I first want to touch on what you mentioned right at the end with alien invasive species. So How would you
Yeah.
apply genetic offsets to alien invasive species and potentially predicting invasion? So I'm doing some work on alien invasive plants. So I'm curious how you would think we can actually apply this to enhance predicting potentially invasive species.
Right. Yeah. So I think there's a lot of opportunity here because we often, have knowledge of where an invader may have originated in its native range, and be able to identify those regions, and then you can develop predictions based on the pattern of adaptation in the native range. You can make a genetic offset prediction for where within the introduced range it should have maximal fitness, right? Based on its historic distribution and relationship with climate. So I think that could be a very interesting application for biological invasions, because particularly for newly emerging invasions where we're trying to, maybe it's not in equilibrium yet, and you're trying to get a handle on where it might spread, what environments it might spread into. Folks have used SDMs for this type of problem a lot, but again, they ignore the fact that the native range has genetic diversity in it, and you can't really treat the native range as just a single entity, but really, different locations from within the native range probably have different pre-adaptation to areas of the introduced range. And genetic offsets would let you look for that. And also, if you have any kind of time series, for example, if you can go into the herbaria and look at, where a species was maybe first introduced, and then how it spread through time, based on documented collections within the herbaria, then you could also look at how well those match genetic offset predictions in terms of it moving into those environments that it's most pre-adapted to.
Yeah, and so to some extent it ties into native range size then as well. So the idea is that the bigger the native range size, the more likely it is to, or the bigger the chance of invading. But that's not always the case. So we've done some work on a Hypericum species recently that comes from a very small native range size, and it's still invading. Okay, those are some great ideas, and I've looked at it to some extent from some of this from a plant reproduction perspective, where we've, for example, measured cogamy, so anthostigma distance, and that's how that changes over time if the pollinators are not available. Anyways, I'll come back to that question and expand on that and link that to what we're doing here. Let me just have a look at hands. And by Dave Berger. Dave, over to you.
Okay, good day. Thanks for the great presentation. My camera's in the wrong position. So I was just wondering, how applicable is your approach? Because the examples you're given in North America where there's massive distances. And so how does it work on smaller spatial scales?
Yeah, that's a good question. It's really going to come down to the scale of how the environment changes through space. So even at small spatial scales, particularly when there's topography, you can generate some pretty steep environmental gradients. And so then the tractability of the modeling method will come down to the spatial resolution that the data are available. So if you've got very fine-grained environmental data available, even if it's at a small spatial scale, then you can make good predictions using this modeling framework. It'll impart, the extent to which you've got strong local adaptation at those smaller spatial scales will also, of course, reflect dispersal and how much gene flow might be homogenizing the allele frequencies at smaller scales versus those big landscape scales. So there's some sort of multiple interacting processes there.
could you use it for altitudinal gradients of a distribution of has that been done?
Absolutely, yeah. We've started looking at that in the red spruce case study that I showed you, because it shows really steep environmental gradients along altitude. And so the challenge we have there is the gridded climate data that are available, for some of these global data sets really aren't at a fine enough resolution to be able to look at altitudinal changes that are occurring over the space of a few kilometers. And so what we're doing now is basically creating our own data by putting out temperature logging devices along the altitudinal gradient, so we can get a finer scale sense of how the environment changes along the gradient. And so you'd need something like that, I think, because the global gridded climate data sets are just too coarse.
Can I follow up with another basic question? Is you always talk about the populations that you've got. So how do you actually define those populations? Because the forests are probably very homogeneous. So how do you break it up? Do you use your genetic data to break it up first into populations?
Yeah, it's a classic question, and an important one. And the real answer is I've been referring to them as populations out of maybe just sort of history and convenience, but of course they're not discrete. As you noted, a lot of these forest stands are more or less continuous. And so really what I mean by population in the context of a forest, for example, is just, an area where we've gone out and we've sampled enough individuals that we can estimate the frequency of the allele in that area. But of course, the genetic exchange is happening on a much more continuous basis. You're absolutely right.
And is your target sort of 10 individuals in a population? Because I noticed that was more or less the numbers you were working with.
Yeah, there's some good studies looking at the sampling design trade-offs between sampling more individuals within a local area versus being able to sample more spatially distinct areas. And if it's a diploid, then each individual provides you 2 copies of each allele, right? One from mom, one from dad. So if you sample 10 individuals, then you've got a diploid sample size of 20 to estimate allele frequencies from, which is not bad, under a binomial distribution. And you tend to do better keeping the sampling, of individuals within an area at around 10 or so, if it allows you to sample more distinct portions of the environmental gradient, because that's really where your power is going to be for estimating those turnover functions. You need lots of different distributed samples along the gradient to really get a good estimate of what that function looks like.
And are you not concerned about genome size? Does that not influence how many individuals you have to sample?
It certainly influences your sequencing approach. So spruces, for example, all of the conifers have just enormous genomes. The spruce genome is 22 gigabases, so 22 billion base pairs. And so for that genome, and much of it is transposable elements and, lots of repetitive regions. So it's not even feasible to put it all together. So for that data set, we targeted just the coding portions of the genome, the exome. And because that's, most likely going to be functionally contributing to the adaptation we're looking for. And so there are ways to sort of focus in on just the portions of the genome, if the genome is really big, that, make the most sense. But it is an important consideration, is you need enough sequencing that you feel like you're really capturing the regions of the genome that might be involved in adaptation. So you do need to sort of have a dense enough sequencing strategy that you're capturing those regions, even if you're not sequencing every base pair.
Great, thanks very much.
You bet. Good questions. Thank you.
Great, thank you very much for that. I see the other hands dropped. There was a hand by Oladayo. Are you still around for your question? And the other hands I see dropped, but apologies, I see we're also over time. But if there's one or two quick questions, I suppose we can still have them.
Yes, I'm still, and apologies about that. My network actually went off.
Oh, great. Okay, you go for it. Yep.
Yes. All right. Thank you for the space. I must comment that the speaker actually spoke well. Yeah, the whole thing is quite informative. I really appreciate this opportunity as well. But then I want to understand if this algorithm can be used to, predict the genetic offset that happens between coexisting species, like lichen, or like two different species that one actually prey on another. I'm particular about species in the case of disease, because I actually study botany, so I specialize on using plant biopesticides to cure some diseases in plants. So I want to understand if this algorithm can actually predict the relationship between these two species that coexist. For instance, we can take the issue of, let's say, for instance, this tobacco mosaic. So at what point can we say when you begin to increase temperature or you begin to decrease temperature as well, where these can become an advantage to the plant particularly? So is it possible this algorithm can predict this kind of relationship between the coexisting species? Thank you.
That is such an interesting and important question. Thank you for asking it. Nobody's looked at it, to be honest. I think that there probably are some opportunities, and it would be really interesting to do. And I think, one of the relationships that you might predict is that the interaction between a plant host and its pathogen, for example, may be mediated by the environment, right? So the plant's ability to resist infection, for example, may relate to the temperature or moisture availability or other things that are affecting the amount of abiotic stress that the plant is under. And so I think that in that sense, genetic offsets, as we've currently been thinking about them, could be used to predict resistance to the pathogen. And really, instead of the responsive variable in the validation test being height or growth, or some of these other examples I gave, your response variable would be infection rate, or some measure of prevalence. And so, but that would be a very plant-focused type of an approach. You could also approach it from the side of the pathogen, because you've basically got two parties interacting, right? And so I think it would be really interesting to actually develop the similar set of predictions from the pathogen's perspective, if you knew how its ability to infect or become virulent may be a function not only of its host, but also the environment, the abiotic environment that the pathogen is existing within. And so you're really talking about a model that we haven't explored yet, but I think that there's a lot of interesting questions there, and I would love to see somebody start working on it.
Thanks, Stephen. Thanks for the
Thank you
for the question. I'm not going to ask my question because it was in similar lines, but in terms of plant pollinator interactions, because I work on pollinators, and that's a similar thing. Then than a pathogen and a plant. So thanks for those ideas. And it's good to know that that's quite a novel space, that there's space to move into that. We are over time, so I am going to wrap it
Absolutely.
up. I see there are no more questions. So thank you once again. Thanks a lot for your time and sharing all of these insights. Really appreciate it. And thanks everyone for listening in. And in South Africa, it's 5:20, so enjoy the evening. And on your side, Stephen, enjoy the rest of your day. Thanks everyone.
Cheers. Thank you. Appreciate it.
Cheers. Thank you very much.